So in this video, we're going to think about one of the classic signalling systems in animal or in human cells, which is that of growth factor signalling. Okay, so just a recap of a little bit from the cell cycle video, if you haven't already watched that. So let's just remind ourselves of what the different stages of cell division might be. So if we start with fertilization, then we stimulate a cell division cycle which is made up of the phases G1, S, G2 and M. And there's a whole other video on the regulation of the cell cycle that you can watch if you want to know more about the cell cycle. So this is cell division, and cells can go through that as many times as they like. But sometimes what cells decide to do is to opt out of that. So from the G0 phase, from the G1 phase, you can opt out of the cell division cycle. So a G0 cell, which is sometimes known as a quiescent cell, is non-dividing. So that's usually a relatively specialist sort of cell. So the classic example would be a neuron, for example. So usually we have this trade-off of you are either dividing and you're relatively non-specialised, or you have stopped dividing and you're a more specialised or differentiated cell. Okay. And that's usually the trade-off. So usually we'd expect going to fertilisation, sperm meets egg, go into the cell division cycle, make lots and lots of cells, and then they start to become more specialised. So they start to become liver cells or skin cells or neurons. But there are occasions where the cells need to be able to go from G0 back into the cell cycle. So that might be, for example, if we need to repair damage. So for example, if you've cut yourself, so you've got a wound on the surface of your skin, obviously that wound needs to heal. And a way that that happens is that the skin cells on the surface, which would normally be G0 cells, they stimulate a process of going back into the cell division cycle so that you repair the damage. If you just waited for the normal cell division cycle and the stem cells, that would take weeks and weeks and weeks for you to get enough cells to reach the surface of the wound. Whereas if you rely on the cells on the surface, then you can get much more rapid repair of the damage. Okay, so if I just show you that, so if you've got the wound, okay, so this is your skin. At the top we have epidermal cells. We then have the dermis, and then really deep layer we have stem cells. So the stem cells are the ones that are going through cell division, and usually those stem cells, they just sort of work their way up, so eventually they make its way to the surface. So that will take weeks and weeks for that to happen. You don't want a gaping hole in your arm for weeks and weeks. So what happens instead is that those epidermal cells on the surface, which are G0 cells, they go back into cell division so they can start to divide and start to put new cells across the surface. Okay, so we do need a mechanism of going from G0 to G1. So I'm going to draw this out as quite a big cell because there's quite a lot of different processes going to be happening in this cascade. It's quite a complicated cascade and there's quite a number of stages in it. So we're going to go through this nice and slowly, okay? But we need to think about before we start about the details, we need to think about what the signal is. So the signal is epidermal growth factor. which is a small protein. So when the cells detect damage, then they will start to release this signal of epidermal growth factor. So they'll start making that small protein. So that is our signal. So we'll just have that as a blob there. Okay. And then we need to think about what's our response going to be. Well, if our response is going to go into the cell cycle, we know from the other video that the cell cycle is controlled by a number of genes, and particularly by the cyclin gene. So the target for our overall pathway is we need to activate the expression of a cyclin gene. Okay, so that's the gene there. We have the open reading frame, which makes the protein, so that's the bit that's going to make the protein, and then we have the promoter, which is the control region, or the switch if you like. Okay, and obviously the cyclin genes are in the nucleus, so we're going to have to think about multiple components or multiple compartments within the cell. Okay, so we've got our signal is epidermal growth factor. That's on the outside of the cell, and it's a small protein, so it can't get inside the cell. It's too big to fit in an ion channel or a transporter. It stays on the outside all the time, so we need to get the signal into the cytosol first of all, and then into the nucleus. Okay, so there's going to be a number of steps to do that. So obviously, to start with, we need some sort of receptor. Okay, so the receptor in this case is imaginatively named, is the EGFR, which is the epidermal growth factor receptor. Okay, so it's got a single transmembrane domain. So there's there's an external portion, there's a single transmembrane domain, and then there's an internal cytosolic portion. Okay, so there's the growth receptor in its resting state. Okay, so then what happens is it binds to epidermal growth factor, and in doing so, there is a conformational change in the protein. Okay, so we've got conformational change. So the protein has just changed shape as a result of binding to its ligand. Okay, so I've shown that with this. So the outside bit was a sort of flat disc, and now that disc has tilted. So the shape of the receptor has changed in response to binding. And what happens in this case is that means that the receptor can what we call dimerize. So instead of having just one receptor, we've now got two proteins that have come together and have bound to each other. So it couldn't bind before because this kind of outside domain was too big. They couldn't get near each other, but because of the conformational change, the two proteins are now able to stick together. So we've got a dimer. Okay, so we've had a change in the receptors already. The EGFR receptor is what's known as a tyrosine kinase. So kinases are proteins that can phosphorylate other proteins, and these ones are able to phosphorylate specifically at the amino acid tyrosine. So what happens in this case is actually the proteins phosphorylate each other. So they can't phosphorylate themselves, but they can phosphorylate each other. So we have after dimerization, we then have trans phosphorylation. So the proteins first of all come together as a dimer, and then they phosphorylate each other at a tyrosine residue. Okay, so we've had quite a big change in the appearance of the protein. So it used to be a protein by itself with no phosphate groups on it. It's now a dimer of proteins with phosphate groups. So this looks quite different in terms of the inside of the cell. And there are now binding sites for other proteins. So there is a protein called SOS, which binds to these phosphorylated tyrosine groups. So it can't bind in the original state. It can only bind after dimerization and phosphorylation. So we've got binding of a new protein in there, and that results in the activation of another protein called RAS. And RAS is what's known as a monomeric G protein. So you might have met G proteins before, and they're in some of my other videos, where you've got an alpha, beta and a gamma subunit. This is a slightly different type of G protein. It's a monomer, it only works by itself, but it has the same property that it is able to exchange GTP for GDP only when it's active. Okay, so when it binds to SOS, when it binds to SOS, we have GTP, GDP exchange. So our protein is able to activate. Okay, so we've now got 3 proteins in the cascade. We've got the growth factor receptor, we've got SOS, and we've got RAS, which is a G protein. And notice that all of this at the moment is happening at the plasma membrane. So RAS is still, it's got a little lipid tail on it, it's associated with the membrane, it can't sort of diffuse into the cytosol. But the next protein in the system, and I'm just going to change colours for this next bit, is a cytosolic protein. So RAS, once it's activated, is able to activate a protein called MAPK. So that's the active version. That's inactive. That's active. So MAPK stands for MAP kinase, which you might think, well, that's a ridiculous name. What on earth does that mean? Let me draw out the rest of the pathway, and then we'll make sense of that name. So remember, kinases are proteins that phosphorylate other proteins. So MAP kinase is able to phosphorylate a protein called MAP kinase. Okay, so MAP kinase in this case is the substrate, and it acted as a kinase, it added a phosphorylated on, so this one, it was a kinase of MAP kinase, which is why it's got 3 kinases. The same thing happens again. So MAP is MAPK equals MAP kinase. So that again we have what's called a phosphorylation cascade. Okay, so the end point is MAP kinase. Okay. MAP kinase got phosphorylated by MAP kinase, and MAP kinase got phosphorylated by MAP kinase. So we've got these 3 levels of phosphorylation. So those proteins all phosphorylate each other. And that's really important because what we end up there is a process of amplification. So each MAP kinase can phosphorylate 1,000 MAP kinases. Each of those can phosphorylate 1,000 MAP kinases. So we end up with going from a weak signal to a very strong signal in the cell. So because we've got these multiple layers of activation, and each one, it's not one-to-one, it's one-to-many, we end up with amplification. So we've now got phosphorylated MAPK, and what that is able to do is to enter the nucleus. So the non-phosphorylated form can't come in. This one, not allowed in. Phosphorylated form is allowed into the nucleus. The nuclear transporters recognise it specifically. Okay, so we've now got MAP kinase in the nucleus. So what's that going to go and do? Well, it's a kinase, so it's going to go and phosphorylate another protein. So it's going to phosphorylate a protein I should put in purple. It's going to phosphorylate a transcription factor. And the transcription factor is MAP. So we've finally got to where the MAP comes from, and that's mitogen activated protein. So we activated a transcription factor, and what we know transcription factors can do is to bind to the promoters of genes. So the target for this is actually a transcription factor that needs to be phosphorylated in order for it to act as a transcription factor, in order for it to bind to the cyclins. So we make cyclins are expressed. And because we've got cyclins, we can then stimulate cell division. Okay, so we've got a number of different steps in there. Okay, so let's just run through it again. We start off with epidermal growth factor, so that's made as a result of damage. Epidermal growth factor binds to its receptor. There is then conformational change of the receptor. So the receptor changes shape. So the first thing is conformational change. That then allows dimerization, which is the second process. Because we've done dimerization, we can have transphosphorylation, so the proteins phosphorylate each other. We can then have SOS comes in, we can then have RAS. So we're starting to get this cascade happening, okay? All the blue steps are at the plasma membrane. We then have a phosphorylation cascade. So we start with MAPKK, that phosphorylates MAPKK, that phosphorylates MAPK in this phosphorylation cascade that gives us amplification. So we go from having quite a weak signal, so only a couple of RAS proteins would be active, but then we make sure that all the MAP kinase proteins in the cell have been activated through this mechanism. We then go to a process of nuclear import, which is dependent on the phosphate group. We can't import unphosphorylated MAP. MAPK phosphorylates a transcription factor, and the transcription factor binds to the promoter. So we activate gene expression of the cyclin, and that we know that cyclins will stimulate the cell cycle. So we've gone from the signal from wounding through to the activation of cell division through quite a number of different stages. So if you've wounded yourself, then this is a good process to happen. This is the right and appropriate process to happen. But sometimes this process does happen inappropriately. So what you can have is a lot of the genes associated with these are also associated with cancer. Okay, so let me just highlight genes that are associated with cancer. If you have mutations in the growth factor receptor, then sometimes you can get cancer. So if the growth factor receptor is on when it shouldn't be, all of this stuff will happen and you'll have cell division, even if you haven't got the epidermal growth factor. So the growth factor receptor, if that goes wrong, that can stimulate cancer. RAS is associated with cancer. If RAS is switched on permanently, all of this stuff will happen, even if we haven't got receptor binding. the MAP kinases are associated with cancer. If they get switched on accidentally, you don't have to have all of this bit happening at the top. If MAP kinase is switched on accidentally, you'll have gene expression and cell division as a result of the process, and this transcription factor as well. If that transcription factor is switched on at the wrong time, then you can have expression of the cyclin and cell division. So we call these cancer-associated genes, if they're normal, so they're unmutated, we call them proto-oncogenes. If they're mutated, we call them oncogenes. So if this pathway gets mutated, if any component in this pathway gets mutated so that the protein is switched on when it shouldn't be, then that is a mutation that we call an oncogenic mutation. And if it's not checked, if there aren't any other mechanisms happening in the cell, then that could lead to the association of cancer, which of course is unregulated cell division. So usually this is a good process to happen. It's all about repairing a wound, but if the process goes wrong, then we can end up having cancer associated mutations.